So now we're going to go back to some talks, and in my introduction, I think part of that was part of it that was missed was my welcoming of the new graduate students for this year. We got 19 of them, and they'll be introduced this afternoon by Leslie. But one of the features of the science retreat is that we have these graduate student lightning talks, and we have 3 this year. They're 5 minutes each, and they're going to give us an overview of the research that they've done. Of course, it's a great exercise for students to participate in a retreat in this fashion. We'll have a 10-minute question and answer session after the 3 talks, which will be done consecutively. So if you've got questions for any of the talks that you're listening to, please enter them into the chat box, and we'll moderate them after the session. So we're going to hear from 3 students in a row. Austin Green will be the first one. He's from the Sekercioglu's lab. Crystal Sue from the Dale lab, and Kaitlyn Ellis will be the last speaker from Sophie Caron's lab. So let's go to Austin Green.
Everybody, thank you very much for tuning into this talk. My name is Austin Green. I work in the Biodiversity and Conservation Ecology Lab, and I'm going to be talking to you guys about research from a recent undergraduate in our lab, Kelsey Barnick, and she was interested in looking at the effects of Interstate Highway 80 on mammalian community composition across the central Wasatch Mountain Range. And so to get right into it, my research and my dissertation is really focused on how wildlife are using the wildland-urban interface that's here in the Salt Lake Valley and the central Wasatch Mountains. And particularly, we're interested in where these animals are going and how they're accessing these different habitats. And so what you see here is a heat map for mountain lion showing areas of high activity and areas of low activity. And what you can see from this is the areas of high activity are concentrated in the north in these protected areas. This is Red Butte Canyon Research Natural Area. And then this is the top of City Creek, which is a protected watershed. And we see this for just about every species that we look at. The activity is concentrated in these northern areas, and then other pockets of high activity are in the higher elevations of the southern canyons. And then almost without a doubt, across every single species we look at, Interstate Highway 80 right here seems to be some sort of barrier connecting these 2 areas of high activity. And so this really was the rationale behind Kelsey asking this question: has the combination of I-80 and the difference in housing density and human influence affected the mammalian community along the central Wasatch Mountain Range? And so to study this, we set up a small scale camera trapping project where we had a good amount of sites both directly north and south of the interstate, and then some reference sites on both the north and south to see if this effect was distance dependent. And really quick, before we go any forward anymore, I want you guys to all notice how close these sites are together. Theoretically, there should be no difference in what we're measuring right here as opposed to what we're measuring right here. It's all very similar habitat, opposed from being on the north of I-80 and on the south of I-80. And so what we see here is the difference in community similarity, and this is measured through the Jaccard index. And so basically we took Red Butte as a reference site for the rest of our sites, and we said, how similar is the mammal community compared across all of these different sites? And so what you can see is there's a stark difference between sites that are on the north and sites that are on the south side of I-80 in terms of what mammal species they are picking up on the camera. And it's actually a pretty significant difference. And so we wanted to start looking into this a little bit more. What was causing this significant difference between community composition on the north and south of I-80? And just to reiterate, these sites are super close together, so there shouldn't be this stark difference between the north and the south. And so we look kind of more deep into this effect, and we can see that housing density was a major factor for why community similarity was differing across the north and south. The more housing density you had, and this is on a log scale, the less your mammal community at your site was similar to the Red Butte reference sites. However, we also saw, and this is kind of the more interesting effect, we also saw that there was a negative effect of I-80. And so although it's represented as a positive trend here, what you're looking at is the log of the distance from I-80 to the camera. And so what that means is the farther away you are from I-80, the more likely your site is going to resemble that was measured at our reference Red Butte site. So that means that not just the sites that were right next to Red Butte looked like Red Butte, but the sites that were really far, the farthest sites south of Red Butte also looked more like Red Butte. And it was the ones right next to the interstate that didn't resemble that mammal community. And so basically, to come back to this question, has the combination of I-80 and the differences in housing density across the north and south affected the mammalian community along the central Wasatch Mountain Range? Well, we did see that community composition did differ across the north and south, and there was a negative association with both housing density and I-80. So although we can't conclusively say that that's the fact, this does provide evidence that having a major interstate highway going directly through a protected area or undeveloped land can alter the community that it is impacting. And so with that, I'd like to thank all the major partners in my dissertation research. This would not be possible without them. I'd like to thank Kelsey for leading the effort on this project, as well as Chan for being a great mentor. I'd like to thank Monty, David, and Kyle. Stoked to have you guys joining us, David and Kyle. And then I'd like to thank Joseph, Sarah, Susan, Jenny, and Danny, Mary, John, Matt, and Zach, who are members of my Wasatch Wildlife Watch community science team that helped directly with this project. And then, of course, I'd like to thank all of you for listening.
Neil, do you want to introduce Crystal?
Yeah, so next talk, lightning talk, is from Crystal Sue, and she is in Colin Dale's lab.
Hello everyone, today I'm going to walk you through my thesis project and show you some very interesting results. Insect-bacterial symbiosis is a classic example of mutualism widely found in nature, and at least 10% of insects have obligate mutualistic bacteria in the symbionts. These mutualistic symbionts can provide their host nutrients that are usually deficient in their diets, as well as protection against heat predators and fungal infections. The genome of the symbionts have been through degenerations over time, and many of them are dependent on the host for survival. This makes insect symbionts very difficult to be cultured under lab conditions, and therefore limits the researchers to study the symbionts' functions and understand how they transit to a mutualistic lifestyle. My research project is aimed at studying the establishment of the obligate mutualistic association in green weevils by creating an artificially engineered laboratory symbiosis between green weevil and a bacterial strain, Sodalis precaptivus, which we call the strain HS. Strain HS is a free-living bacteria that is considered as an environmental precursor of many other Sodalis allied symbionts, including the primary symbionts in green weevils. It is amenable to culture and genetic modification and can persist in adult weevils without harming the host following injections of the HS cells. These characters make the strain HS a good candidate to establish a new symbiotic association. APO is just an abbreviation of the term aposymbiotic. It basically means that the natural symbionts in the wild-type weevils are removed by feeding weevils antibiotics, and all my studies here are using maize weevil specifically as a model. Here are some more details about the green weevil symbiosis. As I said before, one of the major challenges in this field is that these obligate symbionts cannot be cultured, and this applies to green weevil as well. It is known that the symbionts provide tyrosine and phenylalanine to support the growth of a darker and stronger cuticle in weevils. But another critical feature that differentiates mutualists from pathogens is the ability to undertake vertical transmission. It has been a mystery about how they initially established the mutualistic symbiosis and even let them invade into the reproductive tissues and the embryos. I have tried to inject strain HS into adult weevils and failed to induce any vertical transmission. This leads us to consider the timing of putting these bacterial cells in. Since the female weevils inoculate the primary endosymbionts into eggs in the ovary, I started to work on directly injecting the strain HS into eggs to mimic the more natural pathway. Due to the nature of green weevil oviposition, the eggs are hidden in a small cavity that female weevil drilled inside the corn, and the larvae will feed on the endosperm of the grain and will emerge from the grain once they reach adult stage. These features make the egg injections and rearing very difficult to achieve. After a year of trying, we have successfully developed a solid protocol for microinjections that is capable of transferring HS cells into weevil eggs and rearing the injected eggs to adult stage for reproduction. At the bottom left, you can see that the staining of egg plugs in pink little dots on corn, and there's usually one egg underneath the egg plug. And on the right, it shows how injections was done under a phase contrast microscope. Very recently, we have achieved the first de novo vertical transmission of string HS in aposymbiotic weevils, and this discovery will allow us to further understand the genetics behind the vertical transmission. In order to have a better understanding of where the injected bacteria localized inside the weevils, earlier this year, we constructed an engineered HS strain that expressed M. cherry fluorescence. This M. cherry fluorescence strain was already injected into many apo eggs and established a population that yields a 100% vertical transmission rate at the moment. Here are some latest results we have. So here are the comparison between two weevils under normal light and fluorescent light. The left weevil in each photo is the one with fluorescent strain inside of them, and the one on the right is the normal weevil. We can see that the fluorescence is totally detectable in adults simply by putting them under the fluorescent light. And here are photos of female reproductive system dissected from an F1 generation of the injected population. As you can see from the photo on the right, there is a notably high concentration of fluorescent bacterial cells in the ovaries, specifically in the lateral oviduct, which is a supportive evidence for the vertical transmission achieved in our lab. But for sure, more investigations will need to be done to have a better understanding of the whole story. At last, I would like to thank my committee members for their support and our funding agencies. Thank you for listening, and please let me know if you have any questions.
So our final talk, lightning talk, graduate lightning talk, is going to be from Kaitlyn Ellis in the Caron lab.
Hello. My name is Kaitlyn Ellis, and I'm a graduate student in Sophie Caron's lab. And today, I'm going to tell you a little bit about one of my ongoing projects. Learning is an important function of all brains. As a lab, we are really interested in understanding the neuronal connectivity patterns that enable the brain to learn efficiently. To do this, we have turned to the Drosophila mushroom body, an associated brain center that receives most of its input from the olfactory system. The olfactory system of the fly begins with olfactory sensory neurons, which are housed in their antennae. These neurons then project to a brain center called the antennal lobe. Here, these olfactory sensory neurons make connections with second-order projection neurons. And these projection neurons target two higher brain centers: the mushroom body, which is important for learned behavior, and the lateral horn, which is important for innate behavior. We are particularly interested in the connections between these projection neurons and Kaitlyn cells being the neurons of the mushroom body. During her postdoc, Sophie developed a neuronal tracing technique to map Kaitlyn cell input in order to really understand this connectivity pattern. What she found is that Kaitlyn cell input is biased. Some projection neurons are underrepresented. They make significantly fewer connections with Kaitlyn cells and have fewer presynaptic photons. On the other hand, some projection neurons are overrepresented. They make significantly more connections with Kaitlyn cells and have more presynaptic photons. We believe that underrepresentation preserves the innate behaviors that flies have towards a specific subset of odors, while overrepresentation really predisposes specific odors to be learned in many different contexts. And we believe that these biases are reflective of the fly's ecology. In order to test this idea, I am taking a species comparative approach. I predict that as these different species of fly have adapted to different ecological niches, those biases in Kenyan cell input will have shifted to really reflect those ecological niches. I started out by comparing Drosophila melanogaster, a generalist species, to Drosophila seychellia, a species that is specialized on noni fruit. I chose to compare these two species because while they are closely related and they have the same projection neuron types, this makes it easy for me to really make direct comparisons between these two species. However, what is really interesting is that these two species have evolved to live in very different environments. And so what I predict is that perhaps some of the projection neurons that are responding to odors that are specific to those niches have changed in their connectivity rate between these two species. And what I found is that this is indeed the case. For instance, there are some projection neuron types that are making more connections with Kenyan cells in Seychellia than they are in melanogaster. Two of the most significant differences are to the DL2D and DP1L projection neuron types. What is really interesting is that these two projection neuron types respond to odors that are predominant in noni fruit, the host of Seychellia. On the other hand, some of these projection neurons have a higher connectivity rate to Kenyan cells in melanogaster than they do in Seychellia. Two of these projection neuron types are DC1 and DC3. These two projection neuron types respond to citrus odors. And what is interesting about this find is that previously it has been demonstrated that if given the choice between different fruit types, Drosophila melanogaster will prefer to lay their eggs on citrus. So it does appear that these biases in connectivity really do reflect the expected ecology of the fly. And I would like to thank everyone who has helped this project, and I would like to thank all of you for listening.
Okay, thank you very much for those presentations, Austin, Crystal, and Kaitlyn. Of course, lightning talks deserve thunderous applause, so I'm sure that you can hear that now. So thank you again. If you have questions, and there have been some questions in the chat room, please type them in, and Austin is going to moderate the questions. David Pace has indicated that Kaitlyn may not be here, so if she is, then let us know and we'll direct questions to her. Otherwise, questions for Austin and Crystal. Austin?
Yep. So Kaitlyn's unable to join us, but if you still have questions for her, you can still put them in the chat. I'll put them together and send them to her if she has time to answer them on her own time. She can get to that. But we'll just get right into it. The first question will be for Crystal. And oh, wow, I forgot to say who actually asked the question. Hold on. Marlon asked, how many generations have you been able to observe vertical transmission of M. cherry HS in?
Well, can you hear me?
Yes.
So far we only have F1, and it is actually one experiment we are conducting right now to track and see if this vertical transmission is stable over generations. And we are planning to do like at least 3 to 5 generations to see if there is a drop or a lost or a maintain, which is the perfect option.
Awesome. So the next question was for me. Lisa asked, are there any ways for animals to cross the freeway, like tunnels, bridges, and etc.? Yes, there are. So in 2018, they finished construction on a wildlife overpass up by Parley Summit. And so we are definitely looking into that. A lot of my work is applied conservation. So it's more investigating things, looking for correlations that then can be transferred into actual applied conservation and management on the ground. And so a lot of my research is basically kind of tailored around showing the effects of major sources of fragmentation and potentially showing how solutions like an overpass can help. And so that kind of leads in really well to Martin's question, which was, do you have any cameras on the bridge and are animals using that conduit? Yes, we do. And so that we have about like 16 cameras in a bunch of different areas on that overpass to get both individual information from species and just the community-wide aspect. And one thing we're really interested in looking at, and this was thanks to Monty's suggestion from our lab, was basically how traffic across the overpass is changing, or basically is the directionality of the traffic asymmetric. So what we mean by that is, since we're seeing higher habitat usage and higher site abundance at the sites north of I-80, are we then going to, kind of in correlation with that, see more traffic from certain species going north to south as opposed to south to north? And if so, do those patterns seem to match the patterns that we're seeing with distribution? So basically, the animals that we're seeing more often in the north, are they the ones that are traversing the overpass from north to south more often? And if so, that provides a little more evidence that that theoretically could be a solution for us. And so continued monitoring in the future is really important to see if we continue to get these discrepancies in community similarity across the north and south. But also, I don't know if one wildlife overpass in an area roughly 1,000 square kilometers is going to do a ton to help. But if we can show that it has some type of benefit and is in some way helping out, we can at least try to instigate or advocate for additional measures. All right, so let me see if there are any questions that came in while I was shouting those. Let's see. Richard asked me, do you see animals approach the overpass but become hesitant and not cross? And so we actually only had our cameras on photo mode, unfortunately, which meant that we couldn't really see what happened once they got to the overpass. We've now switched that over to video and photo, so we get a better idea of whether they're actually crossing or not. And if so, what's their behavior once they reach the overpass? Because for whatever reason, UDOT illuminates the overpass at night, which is really strange. They have a bunch of floodlights there. So we're not entirely sure how that's affecting it, but we'll be looking into that now moving forward. And then Jack asked, are any sense of which species are dominant contributes to the Jaccard differences? Yes, we have a pretty good idea of which species are moving that basically forward. And it is seeming to be species that are highly concentrated in red butte and found in really low numbers everywhere else. And that's particularly mountain lion. And on the flip side, we see a lot more red fox and coyote on the south, which is also an interesting thing that we're looking into. But yeah, mountain lion, wild turkey, elk seem to be driving the north, whereas bobcat and coyote seem to be dominating the predator community in the south. And let's see, one more question. Sorry, do we have time for one more question, Neil?
Yeah, go ahead, Austin.
Okay, so David asked, is there any effect of rate of traffic on the amount of animals that use the bridge? Don't know yet. That's something that we're really interested in looking at as well. It's hard to get. And if you're talking about rate of traffic across the bridge, we just simply don't have that information yet. We only set up the cameras basically this season, and so we're still having that data come in. If you're talking about rate of traffic across the bridge through like vehicle traffic, that's something we just don't have that information with. UDOTs can sometimes be difficult to work with. And so basically just getting those numbers is something that we need to do to add that into the model, but currently we don't have that. So.
Okay, if you have additional questions, feel free to type them into the chat, because I think Austin is going to collate them and distribute them to the speakers. And thanks again, Austin, Crystal, and Kaitlyn for those presentations. Very nice. Good to hear what you've been up to.